Our next session in our In the Pipeline series is on protecting the muscle. But first, let's hear from Dr. Scavina about this important therapeutic area.
In Duchenne, the absence of the dystrophin protein means the muscle cell membrane is less stable than in a healthy muscle cell. Due to this instability, the muscle can be damaged, as there is less protection when undergoing normal mechanical stress during physical activity. This damage leads to a number of challenges to the muscle cells, including increased inflammation as a response to changes that occur from disruption in the cell membrane, and reduction in the ability of the muscle cells to repair themselves. Over time, the damage and lack of repair leads to the replacement of muscle with fat and fibrosis, or scar tissue. It also leads to muscle cell death. While there are multiple challenges that arise from the lack of dystrophin, it also means that there are many potential ways to treat the disease. These different downstream effects all have the potential to be treated by various compounds to help protect and strengthen the muscle, such as minimizing the amount of damage that occurs from normal muscle use, reducing the amount of fat and fibrosis infiltrating the muscle, improving the capacity of the muscle to repair itself, and minimizing inflammation. By targeting the various downstream effects, we can strengthen and protect the muscle and provide better outcomes for individuals with Duchenne and Becker, regardless of their genetic variant or change.
Thank you, Meina. Joining us for this evening's panel, Bassem El-Mankabadi from Fibrogen, Paolo Batica from Milestone Pharmaco, Alan Russell from Edgewise Therapeutics, and Linda Marban from Capricor Therapeutics. Remember, for this discussion will be followed by an open Q&A. You can submit your questions in the question box underneath the player. We're going to begin with Dr. El-Mankabadi. Bassem?
Hi, thank you, Ryan and Eric, for introducing us here. I'm delighted on behalf of Fibrogen to be a part in this successful mission for PPMD, and we are thankful for you guys to invite us. So I'm going to go ahead and present my slides. So technically what we're talking here right now, let me minimize this, and we're going to go. We are delighted to have phase 3 started in our program. 2 studies for Duchenne muscular dystrophy. The first one is Lelantos 1, which is non-ambulatory patients. The slide that the first trials is the targeting age 12 and older for non-Duchenne muscular dystrophy. We are targeting 90 patients to be enrolled in these trials. The dose of pamrevlumab will be given in 35 milligram per kilogram in biweekly infusion for 52 weeks. And then patients who were randomized to the both arms will be given steroids in both arms. Standard of care is given in both arms. So the deflazacort or prednisone orally will be given in active arm, which the one that with pamrevlumab, and also with the placebo arm. After 1 year of randomization, all patients who decided to roll over into the open label extension trial will be given the pamrevlumab in an open setting, in addition to any approved therapy. So any approved therapy can be given to our patients in the OLE section. That's in regards to the non-ambulatory trial Duchenne. The next slides, please. I'm just saying the slides by memory now. I can't see it. But the primary endpoint is our performance of the upper limb, version 2. Changes from baseline to week 52. Our secondary endpoint divided into 3 categories. Actually, the first one is a pulmonary assessment, will be the change in the % predicted forced vital capacity. and also the PEC expiratory flow from baseline to week 52. The second category here is the performance. We're going to test the strength of the hands, which is the grip strength, that change from baseline to week 52. The third assessment will be the cardiac evaluation, which will be ejection fraction, left ventricular ejection fraction changes from baseline to week 52, assessed by MRI. Next slide, please. That's Lelantos. The safety, of course, will be assessed throughout the trials. And also, we have a bunch of exploratory endpoints, will be the Duchenne video assessment, will be evaluated. The caregivers will record some video for our patients through our vendors that we hired here in FibroGen. That will tell us any significant changes while the patient occurring in the patient disease progression while the patient resting at home. So that will be an exploratory endpoint in addition to the gene LTB-before and dilated cardiomyopathy. And kudos to the FDA. Actually, they are the one who highlighted that to us during our meeting with them. And we are delighted to have it as an exploratory endpoint to study here in our trial. A bunch of MRIs has been moved to the exploratory endpoint, which is the cardiac fibrosis assessed by MRI, in addition to the muscle fibrosis by biceps brachii, especially the biceps brachii muscle fibrosis. That will be assessed by MRI. Next slide, please. So that's a talk. So let's change gear here to that Lelantos 2. Our second trial here is for ambulatory patients who are 6 to less than 12 years of age. The same concept will be a randomized trial for 1 year. And all patients who were decided to roll over into an open label extension will be given pamrevlumab in an open setting in addition to any approved therapy, including even gene therapy once it gets approved. So technically, it's just the placebo part will be the first year of randomization. The dose, as shown here in the slides, will be 35 milligram per kilogram bi-weekly infusion. Of course, the placebo arm will be matching placebo in addition to the steroid PO, deflazacort or systemic steroids. Again, all patients who decided to roll into OLE can be given pamrevlumab in addition to any approved therapy. Next slide. So our primary endpoint actually here is ambulatory assessment will be the North Star from baseline to week 52. Any change in that will be assessed. In a bunch of other ambulatory tests here, it's forced stair climb velocity, time to stand, and loss of ambulation. And again, loss of ambulation has been added based on the EMA feedback to us, and we decided to add it, and we agreed with them, and we changed a little bit in our exclusion criteria for this study specifically to match that, to add that loss of ambulation. So kudos to the EMA as well in that regards. Next slides. All right, in conclusion, so pamrevlumab mechanism of action can mitigate fibrotic changes in the muscles of the Duchenne patients. Pamrevlumab can potentially be used in a broad range of patients regardless of their genetic mutation. We don't have a specific genetic mutation to be enrolled in our trial. So any patients, all comers can come in. Our phase 3 clinical trials are evaluating the efficacy and safety of pamrevlumab are ongoing right now. And we are delighted to enroll a bunch of patients in these 2 trials. And we are on track on track to complete enrollment by the end of this year, hopefully mid next year. So again, thank you on behalf of FibroGen to invite FibroGen to present our milestone here for phase 3 clinical trials. If you have any question, the next slide showing my email, my team email, lelantos@fibrogen.com. Feel free to reach out to us at any time. We can help you to enroll. Thank you very much, PPMD.
Thank you, Bassem. I really appreciate that. One quick follow-up question. Are you thinking that these 2 studies will be sufficient to drive an NDA submission for regulatory approval?
Excellent question, and the answer is yes, and that's exactly what we agreed upon with our meetings with the health authorities. 2 trials, one in ambulatory, one in non-ambulatory, can be sufficient for BLA.
Wonderful. Thank you so much. Okay, we're going to turn to our next speaker, Alan Russell from Edgewise Therapeutics. You're up next.
Thanks very much. Thank you. For those astute people around me, I am not Joanne Donovan, our Chief Medical Officer. I'm Alan Russell, Chief Scientific Officer. She is my better, but you have to put up with me for a bit. And thanks a lot to PPMD for the invitation. We're really excited to tell you about a new therapy that's currently in early clinical trials, 5506, which is a protective muscle therapy for different types of muscular dystrophy patients. Next slide. This is our forward-looking statement. We are a public company, so we're obliged to put this up just to say that I will be making reference to events that haven't happened yet, and also to note that 5506 is an investigational therapy that has not been approved in any territory. Next slide. So in muscular dystrophy, when dystrophin is deficient or absent, such as in Duchenne or Becker muscular dystrophy, the stress of everyday use of muscles triggers muscle damage that eventually leads to loss of function. The myofibrils that make up muscle have important proteins in them called myosin. The interaction of myosin with other proteins causes muscle contraction, just the normal activity of muscle, and delivers power of muscles to conduct everyday activities. And I'll tell you why this is relevant in a couple of slides. Next slide. So our approach is to protect those most susceptible muscle fibers in different types of muscular dystrophy, the exemplars being Becker and Duchenne. So we are made of fast and slow skeletal muscle fibers. In dystrophic muscle, the fast muscle fibers are more susceptible to damage due to the lack of dystrophin. Okay, if you're interested in learning a little bit more about that, you can see my face talking again in the voluntary section. So within the PPMD website, there's me talking again in a more detailed way about these. We've designed an investigational therapy, 5506, that protects these fast muscle fibers. Your slow muscle fibers are not directly affected by 5506. And in animal models of muscular dystrophy, 5506 protects those fast fibers from injury, improving strength, limiting muscle wasting, and the long-term development of fibrosis, including cardiomyopathy. We believe that protecting fast fibers may prevent further muscle degeneration and slow progression of disease. A phase 1 trial is currently ongoing in unaffected adults. I'll tell you a little bit more about that and a little bit more about the target in the next slide. Next slide, please. So I mentioned this protein myosin, which is important for force development in muscle. In that graphic on the left-hand side, it makes that middle strip of the muscle. 5506 is specifically designed to selectively inhibit the myosin from fast fibers only. Okay, our goal is to limit myosin to a level that prevents breakdown, but not healthy contraction. So we hope to limit those susceptible fast fibers from injuring themselves, not affect the slow fibers, and not affect healthy contraction. That should allow you to do everyday activities, but without injuring the muscle. In animal models of dystrophinopathy, 5506 results in decreased CK and other biomarkers, as well as increased activity. And I'll show you just one little snippet of our preclinical data on the next slide. So we have done a lot of preclinical studies in different models, but probably the most complicated model that you can run is the dystrophic dog. You've seen much of this data with the AAV microdystrophins, for instance. In this case, what we did is we took symptomatic dogs with Duchenne muscular dystrophy, and we gave them short courses of either vehicle or 5506. And you can see two effects of the compound in the graphs below. So as you probably know, in Duchenne, the muscle injury biomarker creatine kinase is increased, and that's usually believed to be as a result of muscle damage. What we saw when we dosed the dogs with 5506 is a decrease in CK. We also observed a really interesting increase in habitual activity in these affected dogs. So these are pretty old dogs, and we saw an approximately 30% increase in habitual activity during the dosing period. So a combination of reduced injury and increased activity. Next slide. So this is just a review of what we have in 5506. It's a small molecule, once a day tablet or liquid suspension. Now, because we're taking a kind of mechanical approach to the disease, we're potentially appropriate for any age or any mutation in Becker or Duchenne muscular dystrophy. As I mentioned, a phase 1 study is ongoing in unaffected adults, and we're also enrolling patients with Becker for a short study to measure biomarkers and safety as part of that phase 1 study. We're currently planning a phase 2 study in adults and adolescents with Becker, as well as an open label extension to study those folks late down the road to begin next year. And we're planning studies of boys with Duchenne that we hope to start next year. As I mentioned, this structural approach potentially could also be used beyond Duchenne and Becker into some of the limb-girdle and potentially other muscular dystrophies. Next slide. So thanks so much for your time. If you have any questions or comments, please contact the email below, info@edgewise.com. Thank you very much.
Great. Thank you so much, Alan. One brief follow-on question. As you're considering potential ways to measure efficacy, are you thinking about muscle MRI given the mechanism of your drug?
Yes. With any protective therapy, we're really interested in muscle injury biomarkers, so things like creatine kinase, right? But as you know, the world of MRI is really progressing very fast, particularly with the wonderful work done by the Imaging DMD folks over in Florida, and we hope to tap into that actively to look at changes over time with that biomarker.
Wonderful. Thank you so much, and it's always great to see new companies entering the space and introducing them to the community. It's wonderful. So we're going to turn to our next speaker, Paolo Batica from Pharmaco, who is ready to talk about Juvenistat.
Well, thanks PPMD for allowing Pharmaco to update the community on the status of the development of Juvenistat in Duchenne. First of all, my disclaimer, I'm a full-time employee of Pharmaco, that is the manufacturer of Juvenistat. Juvenistat is currently in development for the treatment of Duchenne and Becker muscular dystrophies. It is not approved for sale in any country around the world, including the United States. And this presentation is really intended for dissemination and discussion of scientific information only. So, as you are now well aware, the lack of dystrophin, which is the key feature of Duchenne muscular dystrophy, is associated with a much more frequent injury and damage of the muscle cells. Not only that, the lack of dystrophin is also associated with an impairment in the repairing mechanism. Juvenistat, which is a pan-HDAC inhibitor, is actually working by activating this muscle repair mechanism. And in particular, it reduces inflammation, it reduces fat and fibrosis replacement, and it increases muscle regeneration. So as a result of all this activity, in essence, Juvenistat counteracts all the events which are triggered in the muscle and in the outside matrix that are triggered by the lack of dystrophin. And these are all the processes that eventually lead to the muscle replacement by fat and by scars. And we actually have shown these effects not only in the preclinical studies, but actually also in the main part of our phase 2 study in Duchenne boys. In the Duchenne boys, we were able to show that 1 year of treatment of Juvenistat on top of steroids led to an increase in muscle fiber diameter, an increase in regenerative muscle fibers, and a decrease in tissue damage, inflammation, and fat replacement. The kids that actually were part of this study were asked to continue treatment, and all of them actually agreed to. And as a result of that, we have now 18 kids that have been on treatment with Juvenile Stata on top of steroids for more than 7 years. So we can now look at what Juvenile Stata might be doing on what we call the disease milestone. And also we can look at whether the counteracting of the muscle deterioration that I was mentioning before and that was shown in the histology could also be replicated in the MRI. The result is yes, we were able to show that 2 years of treatment with Juvenile Stata on top of steroids was reduced to, was associated to reduced fat infiltration in the thigh muscle measured by MRI. And also we were able to show in these studies after more than 7 years of treatment that the disease progression is actually delayed by Juvenile Stata, and in particular, the age at loss of ambulation was actually, the mean age was 16 years compared to the 13.5 years that has been published by the Synergy group a couple of years ago. We were also able to show in these studies and this patient that we can delay the decline in respiratory function. And if you want more details on all these studies, please go to the Italfarmaco on demand video. Well, where are we in the development of Juvenile Stata? As you are probably aware, we are conducting a phase 3 pivotal study. The objective of the study is to demonstrate that Juvenile Stata can preserve muscle mass and delay the disease progression. And this is done by making a number of assessments throughout the study. The Forster climb is a primary endpoint of the study. And then we are actually conducting all the standard tests that you are probably very familiar with by now, such as the North Star, the time to rise, the 6-minute walk test. We are measuring strength. We are measuring the fatty infiltration by quantitative MRI. We are measuring pulmonary function and heart function. The design of the study is depicted there. So at the baseline, the kids who are 6 years and older on stable corticosteroids and able to walk are randomized to either Juvenile Stata or placebo in a 2-to-1 ratio. And they are then being followed for 18 months. And throughout the 18 months, we do all the assessment that I've listed before. And at the end of the study, then the kids that want to participate are allowed to move to the next study, which I'm going to show now, which is in essence a long-term safety study where all kids are receiving Juvenile Stata, so there is no placebo here. And the study is objective is to evaluate the safety and tolerability of Juvenile Stata in the long term, but also to evaluate the long-term effect on the muscle function and strength. And depending on whether the participants are ambulatory or not, we are obviously measuring different tests, like the one which I mentioned before, 6-minute walk test, North Star, and Forster climb for the ambulant boys. For the non-ambulant boys, we are measuring the performance of the upper limb. So with regards to the regulatory path, you may recall that we had a webinar some time ago saying that the futility was actually passed. So we had an interim analysis to look at whether the study was futile. The answer from the independent data monitoring committee was that the study was not futile and so enrollment could continue. And last September, we have completed recruitment with 179 boys recruited, and we are expecting the final data by the end of second quarter 2022. So this slide is really to say that there are different developments that we are doing in muscular dystrophies. On one hand, Juvenile Stata is being tested not only in Duchenne, but also in Becker. And I will be presenting the top line results on Saturday. And we also have some discovery programs that hopefully will provide new molecules that can then move to the clinic soon. And with that, I would like really to thank all the DMD boys that took part in our studies and their families, and obviously also the Becker subject that took part in the phase 2 study, and the clinical sites that did an excellent job to carry on the studies, especially in this hard time of pandemic. And last but not least, all the patients' organizations which are listed there that have actually supported quite a lot. For any information you might want about the studies, please go to the clinicaltrials.gov with the code that you see there. And obviously, the other possibility is to send an email to the patient advocacy at italfarmaco.com. Thanks a lot.
Do you want to ask a follow-on
question?
Yeah.
Sure. So as you've kind of moved past, could you give us some insight into kind of how many patients or you've had that have rolled over into your open label as you're kind of looking towards your 2023 data?
So we now have probably around 130, 140 patients that have completed the EPD study, and all of them except like 10 have rolled over into the open label study.
That's great. Thank you.
Wonderful. You know, we are going to figure out how to make this up to you, Paolo. I very much apologize for the technical difficulties. We're going to move to our next speaker now is Linda Marban from Capricor. Linda, are you ready?
I'm ready.
Wonderful.
Can you show my slides, please, now? Thank you. We are developing a cell-based therapeutic called CAP-1002 to use in later stage DMD. I would like to thank PPMD for the opportunity to present to you today. Next slide, please. Capricor is a publicly traded, NASDAQ-listed company under the symbol CAPR, and this is our forward-looking statement slide. Next slide, please. CAP-1002 is an off-the-shelf cell-based therapeutic that is not a stem cell. It works by releasing exosomes after the cells are infused into the body. The exosomes travel to the site of injury and are immunomodulatory and anti-inflammatory, and also drive muscle cells back into the cell cycle, causing new muscle cells to be formed. Most importantly, they have been used in over 200 people in multiple clinical trials, and so far have shown no adverse safety signals and encouraging efficacy signals in treating all kinds of muscle dysfunction, including heart disease. We have conducted 2 clinical trials in DMD, HOPE Duchenne and HOPE 2. Both were focusing on addressing the cardiac and skeletal muscle function in later stage, primarily non-ambulant DMD subjects. HOPE 2 was a randomized, double-blind, placebo-controlled clinical trial open for enrollment at 9 DMD certified sites. We treated 20 patients in this clinical trial. Next slide. We have previously released preliminary top-line data shown here. The final data set has now been submitted for peer-reviewed publication. As previously presented, we had strong signals of efficacy. Shown here is the data from the PUL 2.0, which measures the function of the shoulders, arms, and hands, and is used primarily as a metric of disease progression in those that are non-ambulant. We see a 2.4-point difference in decline over 12 months in those treated with CAP-1002 versus those that received placebo, which could suggest delaying disease progression by greater than 1 year. FDA has said that they will accept the PUL as a primary efficacy endpoint for approval for CAP-1002. Next slide, please. We also saw marked improvements in cardiac function as measured by MRI. Ejection fraction is a gold standard measure of how the heart is functioning to meet the needs of the body. And what we see here for the first time is a statistically significant improvement in ejection fraction in those treated with CAP-1002 compared to placebo. This type of delay of progression in heart disease could be the first therapeutic in the DMD space that actually improves heart function, the decline of which can lead to great morbidity and even death. We also had a statistically significant improvement in the levels of CKMB, which is a validated biomarker of cardiac muscle cell damage. We are now making CAP-1002 available to all eligible participants in HOPE-2 in an open label extension study. Next slide, please. Shown here is a slide from a survey conducted a few years ago on DMD patients. As you can see on both of the patients and the parents panel, the ability to lean forward and sit back up is a critical component of quality of life. And in case you can't see it very well, it is shown as number 7 on the patient side and number 8 on the parent side. Next slide, please. In HOPE-2 open label extension, we decided to tackle the issue of clinical meaningfulness by following the patients using a novel measurement tool developed by Casimir, which was to measure the function of the patients living their own lives. Casimir uses in-home video assessments called DVA, custom designed by each patient and their family to track disease progression or attenuation with a therapeutic. Most of the patients in the open label extension study of HOPE-2 are participating in this Casimir substudy. Next slide, please.
We are recording Carter with a stuffed turtle under his left knee because it's painful for him to have his legs like this. So when I say go, follow my instructions as I give them. If you get stuck, just say help and I will stop filming to help you. Ready?
And tell me when
go.
it's done.
Sit up straight. Lean forward and then go back to sitting up straight.
Now we're going to play the second video.
When I say go, follow my instructions as I give them. If you get stuck, just say help and I will stop filming to help you. Ready, go. Sit up straight. Lean forward and then go back to sitting upright.
Okay, and we're back, and we're going to open it up for panel discussions. Thank you so much, Linda. Linda, did you have one more comment on that?
I do. I just want to say that it's now able to lean forward and sit back up. As I showed you from the survey, this is a very important component of quality of life. There are other videos that we are currently reviewing, and we will be presenting those to FDA as validation of the clinical effectiveness of CAP-1002. What we know is that people with DMD don't get better on their own over time. But mostly what I want to say is thank you to Carter, the young man who opened the doors to his life to let us see what he's doing there, and to all of the patients in our trials, past, present, and future, that are willing to let us study them to make the world better for all of you. So thank you.
Great.
Now you can open it for questions.
Thank you, Linda. And one clarifying question to that video. Can you just clarify how many weeks from baseline that second video was taken, given that there was a break in the time that they were on therapy again?
So the patient, he was videoed at baseline. That was the first video. Then he came in for his infusion. And then that was 10 to 12 weeks after the first infusion and just prior to the second infusion. So they're getting 4 infusions over time.
Okay.
Over 1 year.
Thank you so much. All right, Eric's going to open us up now for panel Q&A.
Yeah. So this first one that came in, and I'm going to direct this to both Paolo and Alan. Paolo, you've obviously already had some experience in this area, and Alan, it's going to be something that obviously Edgewise is thinking about. But it's with, in the space of a clinical trial, defining Duchenne versus Becker and how you're thinking about that. Is it just related to the mutation? Are you doing it with mutation and function? Age a component? Just kind of walk us through kind of what you've been defining within your trials for those, kind of separating those two populations. And Paolo, I'll start with you and what.
So, yeah. So as far as the inclusion in the 2 different studies for Duchenne, the inclusion was based on the diagnosis of Duchenne, the genetic diagnosis of Duchenne. the age was 6 and above. They, as I said, they could come into the study regardless of the genetic defect, so all comers. And they actually had to be on corticosteroids for a stable 6 months. Plus, they had some inclusion based on function at the beginning. For Becker, also here it was all comers. It was based on genetic diagnosis also in this case. And here we allowed patients that were adults, so 18 to 65. And they had to have a relatively maintained function based on a 6-minute walk distance between 200 and 450 meters.
Great, thank you. And
No problem.
Alan, how are you all kind of thinking about this?
So for our phase 1 study, we're including all individuals with a Becker diagnosis. There are limits based on age and functional status, which are fairly basic, but we're taking a pan approach at this stage.
Great, thank you. Basim, one of the things that you showed at the end was in your open label extension, patients are able to add on other approved therapies, which we're really excited about as we've talked about combination therapies. We do think is going to be a big way forward. Now, with that, if those patients are on taking pamrevlumab as well as an exon skipping therapy, that's a lot of infusions. Are you allowing the use of ports within kind of the open label extension as just to kind of decrease burden on families?
Absolutely, that's an excellent question. Yeah, Portocast is allowed, and FibroGen will going to take care of the cost of this Portocast if needed. We left it for the PI discretion whether the patient can get the vein access, and we'll take it in a case by case, but we will take care of that. In terms of the infusion, we ask the investigator to just wait for 3 hours between any infusion. Once the patient in the OLE, they get pamrevlumab first and flush the line, then wait 3 hours, get the other infusion. It will be a long study visit, however, it's manageable.
Great, no, thank you. These are, definitely challenges that we're gonna face in the future, but those are kind of positive challenges if we have multiple therapies available. Linda, during that period where there was a little bit of a gap in the trial, that kind of a 9-month, were you able to continue following those patients and kind of learn about the changes from those who were able to be on therapy and then kind of gap in back onto CAP-1002?
Oh, no, we actually did not follow the patients during the gap, but what we'll have is their last study visit and then their baseline visit in the open label extension. And we're actually looking at that data right now. It's really exciting data. And what I can tell you is that we have an expanded access program that's going into place for our people in open label extension, because we're getting so many calls from people who want to be able to continue with CAP-1002. And so, the message itself is that the families are definitely seeing a difference in how their kids are feeling and functioning.
Great, thank you. Alan, as you have talked about with Edgewise's product aiming to protect fast twitch muscles, is this going to be a therapy where especially early treatment and early intervention is going to be more beneficial? And is there potentially a threshold as we do accumulate, fat and fibrosis over time, where there might be an impact on the efficacy of the therapy?
Yeah, that's a really good question. I think with all interventions, the earlier that you intervene, the bigger the signal that you can have, right? But even in later stage individuals with Duchenne or Becker, they still have muscle, and there's no reason that we can't protect that muscle. The dose response may be different, but we fully intend to explore the potential for the compound in all types of patients.
Great, thank you. And Paulo, so I know that although that slide was a little bit off, it did show that there was, from your phase 2 and your open label extension, that with the addition of steroids, there was some additional delaying of disease milestones like loss of ambulation. Have you been able to explore any respiratory or cardiac function as well, or plans to track that in the future to see how those disease milestones may also be potentially delayed with the use of givinostat?
So, Eric, first of all, let me tell obviously there is a on-demand video which is actually telling the whole story. So if anybody wants to go and look at that, they will see the full data set. The quick answer is yes. We have been looking at the respiratory function regularly. And what we are seeing is that if we look at the forced vital capacity percentage predicted, the decline is approximately 2% per year. And you probably know that based on natural history, we are usually talking about 5% loss per year. And if we look at the PEF, so the peak expiratory flow, which is in essence an assessment of the respiratory muscle function, while in the natural history you see also here a decline of approximately 5% per year, we see no decline. And this is, as I said, in the 8th year of treatment with Juvenostat on top of steroids.
Great, thank you. And this question for Alan, with Edgewise, as you're looking, can you, I guess, explain a little more what kind of biomarkers that you're going to be looking at within the smaller study where with the Becker patients are being included? I know it's largely with some healthy volunteers, but what all Edgewise is kind of interested in trying to capture?
I think most of us are familiar with creatine kinase. That's a biomarker that's been talked about a lot. I'm personally a fan of looking at aggregate biomarkers of muscle injury. So we're looking at other biomarkers such as myoglobin, such as the troponins. We published a story on the troponin subunits secreted into circulation in Duchenne and Becker patients in muscle and nerve this year, which is kind of interesting. And we're also relying quite heavily on the SomaScan strategy that you guys really took forward first. In Duchenne. They've now expanded that up to 7,000 biomarkers, and we're doing some really interesting studies right now to explore the role of proteomics to give you an idea of muscle injury.
Great. Can I ask a follow-on question in regards to this population of Becker? Well, first I want to say that you're all looking at patients with high unmet need, and I want to thank you for that. Whether it's non-ambulatory patients or Becker patients, these are badly needed for clinical trials, et cetera. For Edgewise and Intel Pharmaco, and we start with Paolo, can you talk about the Becker populations in terms of the functional endpoints that are being used and being explored in the future? The fact that we may not have as robust natural history data and sort of the challenges with this population overall?
So, Ryan, you're touching a very important point because clearly in the Becker population, on the wealth of knowledge that we have in Duchenne by now is not there. As you will hear on Saturday, obviously the study we have conducted was in essence an exploratory study, not only looking at what Juvenostat is doing in this patient population, but also learning more about how one can actually conduct a study in a Becker population. Mm-hmm. I mean, my first impression, and we really have preliminary data, top-line data at the moment, and we'll have to do much more analysis, but it looks like all the tests we are very much used to, like the 6-minute walk test or the time function test, may not be ideal actually in this population. We have seen in our study, this is something that we have read many times, there is a lot of heterogeneity. So these are all factors that certainly will count when conducting a clinical study.
Yes. And Alan?
Yeah, it's, being able to explore the approach in Becker is really a great pleasure. And we've had a few different focus sessions with Becker individuals, and it's been such a joy hearing about their stories and how much, in many ways, they feel underserved by us, essentially the industry or community. As Paolo said, it's a highly variable myopathy, right? However, studies have been published recently looking at longitudinal history of change in these in Becker, particularly that of Luca Bello And it does appear that you can potentially ring fence individuals that may be more likely to progress. We're hoping to get a lot more information on that by conducting a natural history study in Becker individuals. So we'll be kicking that off in the near future. That will hopefully inform endpoints for medicines and approval. But it is an interesting area, and we really want to take our time to find the best ones.
Great. Well, thank you all for joining us this evening. We really appreciate the discussion. We're going to move on to our next Pat Chat, where Pat is going to be joined by Dr. Krista Vandenborn from the University of Florida for a deeper discussion on muscle and imaging. So, Pat.
Thanks, Eric. I'm really thrilled to be speaking to Krista tonight, and really appreciate you taking time, Krista, from your busy schedule. I know imaging has kept you out of trouble these days, right? So first of all, congratulations on your renewal by the NIH for imaging. It's such an important study. So I'd love for you to just lay the groundwork of imaging. I often think of imaging as the 3 sites in the US. Tell me about those 3 sites and how you've thought about how you have expanded your interest way beyond the United States.
Hi, Pat. First of all, thank you very much for spending some time with me today. And I'm just delighted to be part of the PPMD conference again. I really appreciate it. So imaging DMD has grown a lot over the last 10 years. As you know, we started out with 3 imaging sites. There was our partners in Portland, Oregon Health Science University, Children's Hospital of Philadelphia, and then University of Florida. But we've really expanded very broadly. We've really tried very hard to move a lot of these measurements across many different sites. And at this point, we have about 30 imaging sites across the United States and Europe that are collecting data in the same way as we have in our original study. So a lot of expansion, and really with the goal to be able to run and introduce MR imaging into clinical trials.
Yeah, thank you, Krista. I know so many families over those 10 years that have participated twice a year, and really, first of all, look forward to your wonderful staff in participating. A couple things. I know at first when you started out, you were imaging the quadriceps muscle and basically the lower leg, and now you're looking at many more muscles, the trunk, the upper arms. Are you seeing differences, significant differences in how these muscles are changing over time?
That's a very good question, Pat. We've learned a lot, as you know, by having the opportunity to work with so many families that really helped us understand the disease and how the disease affects different muscles. So whereas we collected about 10-year natural history data in the lower extremity muscles, we've expanded to the upper extremity muscles. We're also measuring the respiratory muscles, particularly as the boys get older, we know that it impacts their pulmonary function. And so we really need better measurements there as well. What we've learned is that this pattern of fat infiltration happens across the different muscles, but the rate at which it happens is different. The reason that's really important is that, as you know, we're trying to use MR as potential biomarkers in clinical trials. So if we're looking at younger age groups, then maybe the quadriceps is a better muscle to target. But if we're looking at older boys that are non-ambulant, then there's other muscles such as respiratory muscles or the upper extremity muscles that may be much more valuable. But at the end of the day, what's really important is that this measurement of fat infiltration seems to be a really good measure of disease progression and a valuable endpoint that we can potentially use in clinical trials.
So I think that you're on the process to qualify fat fraction as a biomarker. So are you qualifying, so where are you in the process? And second question is, in terms of, are you qualifying that in a particular muscle or, across the board in terms of many muscles?
Yeah. So what we're doing right now is we're really building on all of the natural history data that we have collected. Our richest data set are in the lower extremity muscles. So for our efforts to move forward in MR biomarker development and qualification, it's really important that you have a rich data set. So our focus has been on fat fraction in 2 lower extremity muscles. That's the vastus lateralis, which is one of the quadriceps muscles, and then the soleus, which is one of the lower leg muscles. So we put together a steering committee, which is an MR imaging steering committee, which has representatives from PPMD, from industry, from NIH, and from academia, really working together to understand the data, and then collaboration with CPAD, that is our partner to look at the regulatory component. We're submitting an application, a letter of intent right now to the FDA. So a lot of work has gone into it, and our primary focus right now is the lower extremity muscles, just because that's the richest data set that we have.
I see. So Critical Path has the Drisk Consortium. So this is a consortia that has a number of data sets within it, looking at disease progression models and trial simulation tools. So can you tell me a little bit about the path to qualify this? Is it, I'm sure it's very complicated because it's an FDA qualification, and so that takes a period of time. But at the end of the day, when we're looking at gene therapy as an example, one of the questions is, how are you going to get that approved, and what does it mean in muscle, and how will we know? Because obviously it was based on people who were doing very well from birth to their very older ages. So this microdystrophin will be hard to figure out unless we have such a validated tool to say the fat fraction has not changed, or has changed, or the rate of change. So can you give us a timeline of that, Krista, and what you see into the future?
Yeah. You're absolutely right, Pat. It's a complex process. It's been a lengthy process. So I'm certainly by no means the expert in regulatory approval, and our team is learning as we go as well. But certainly our partnership with Critical Path and Drisk has been really, really important. They have a lot of experience in this area. So our first step is a submission of a letter of intent, which has been pretty much completed and is ready for submission to the FDA. At that point, we hope to hear back from them. So what we're actually submitting is a letter of intent for a fit-for-purpose application that allows us to really submit data and submit a briefing document that describes the trajectory, the disease trajectory of fat fraction and its relationship to function. What has been really important in the work is not just that MR is sensitive and it tracks disease progression, but ultimately that it is closely related or is associated with functional endpoints, and it can predict future function. So in collaboration with Bill Rooney and Sarah Kim and others, we have really invested a lot of effort to understanding how well MR biomarkers predict future changes in function. And that's ultimately really important, because the MR measurements will tell us, hopefully quicker, that a drug may be working or not, but you ultimately want to know that that has functional implications. And that's really what the goal is of this project.
Great. So, and Krista, the study is still open for people to join this study, is that right?
So right now, the study was renewed over the last year. We have changed the direction a little bit in that the sense that we will continue to develop these measurements. We're focusing now more on respiratory muscles in non-ambulant patients. As you know, that is a group that particularly is in need of more outcome measures. And then, and follow up to some of the discussions earlier, we're also looking at Becker's patients. There is a real need to understand the natural history of Becker's as well as having more measurements. So that will be the focus. So we've expanded. We are looking at both Duchenne as well as Becker's patients.
And you're also including female manifesting carriers?
Yes. We are doing a separate small study that is going to focus, thanks to your support, Pat. We really appreciate that. We are going to be looking at female carriers. We've done a little bit work, but we're going to expand on that as well.
Good. Krista, I'm really so thrilled about imaging DMD and what it can bring to this community. A highly sensitive biomarker that really can predict function, that isn't biased, right? That isn't based on someone's opinion. And I think that your work in both ambulatory and non-ambulatory and Becker, and certainly manifesting carriers, is remarkable. So we're thrilled to be a part of imaging, and for anyone listening, joining an imaging study I think is so critical to progress. I can't thank you enough for spending time with us tonight. We so appreciate all of you, and your team is such a great team. It's a pleasure to work with you. And so next year we'll be together in Scottsdale. So thank you very much for joining us tonight, Krista.
Thank you, and thank you to all of the families.
Thank you.